Dear students, today I'll be talking about germplasm conservation. Now, this germplasm broadly refers to the hereditary material transmitted to the offspring. Now, this conservation of germplasm play an important role in all breeding programs. Saving selected seeds or vegetative propagules from one season to the next one is regarded as primitive but conventional germplasm preservation and management and is highly valuable in breeding programs. Now, the very objective of the germplasm conservation or storage is to preserve the genetic diversity of a particular plant or genetic stock for its use at any time in future. Now, in recent years, many new plants species with desired and improved characteristics have started replacing the primitive and conventionally used agriculture plants. Now, it is important to conserve the endangered plant and or else some of the valuable genetic trait present in the primitive plant may be lost. So these are the reasons why we require germplasm conservation. Now, there are two approaches for germplasm conservation of plant genetic material. It's in situ conservation or ex situ conservation. Now, what is in situ conservation? In situ conservation means the conservation of germplasm in their natural environment. That is by establishing biosphere reserves, maybe, or natural national parks or gene sanctuaries, all this. And all these are part of in situ conservation. So now this approach is particularly useful for preservation of land plants in a near natural habitat along with several wild relatives with genetic diversity. Now, the in situ conservation is considered as a high priority germplasm preservation program. But the major limitations of in situ preservation is that the risk of losing germplasm is always there because of environmental hazards. And also the cost of maintenance of a large number of genotypes is very high. Now, the other one is ex situ conservation. Now, ex situ conservation is a chief method for the preservation of germplasm obtained from cultivated and wild plant material. The genetic material in the form of seeds or from in vitro culture can be preserved as gene bank for long-term storage under suitable conditions. For successful establishment of gene banks, adequate knowledge of genetic structure of plant populations and the technique involved in sampling, regeneration, and maintenance are essential. Now, gene banks are represented as in vivo and in vitro gene banks. Now, banks in which genetic resources are preserved by conventional method, for example, seeds, propagules, etc., are called as in vivo gene banks, whereas banks in which genetic resources are preserved by non-conventional methods, such as cell and tissue culture methods, these are called in vitro gene banks. Now, both these methods ensure the availability of valuable germplasm to a breeder to develop new and improved varieties. Now, generally, as I said, this germplasm conservation is in the form of seeds. So usually seed are the most common and convenient material to conserve the germplasm. And this is because many plants are propagated through seeds and seeds occupy a relatively small space. Further, seeds can be easily transported to various places. However, there are certain limitations in the conservation of seeds, which includes that the viability of seeds is reduced or lost with the passage of time. The seeds are susceptible to insects or pathogen attack, often leading to their destruction. And it is difficult to maintain clones through seed conservation. And in certain cases, the seeds are heterogeneous and therefore they are not suitable for true genotype maintenance. So we have in vitro method for germplasm conservation. Now, in vitro method employs the conserving shoots, meristems, embryos, and other vegetative parts of the vegetatively propagated plants. Now, there are several advantages of using in vitro germplasm conservation process. That is, large quantities of materials can be preserved in small space. Now, the germplasm preserved can be maintained in an environment free from pathogens. It can be protected from nature's hazards. And from the germplasm stock, a large number of plants can be obtained whenever it is required. And apart from that, obstacles for their transport through national and international borders are also minimal. So these are the reasons why we prefer in vitro germplasm conservation method over the conventional method of germplasm conservation. So there are mainly three approaches for the in vitro conservation of germplasm. It can be cryopreservation, it can be cold storage, or it can be a low pressure, low oxygen storage. So the most important being cryopreservation. So what exactly is cryopreservation? Cryopreservation literally means preservation in the frozen state. Now the principle involved in cryopreservation is to bring the plant cell and tissue to a zero metabolism or a non-dividing states by reducing the temperature in the presence of cryoprotectants. So we use cryoprotectants like glycerol, DMSO, etc. And we keep our cells at minus 196 degrees Celsius in liquid nitrogen. So at the temperature of liquid nitrogen, that is at minus 196 degrees Celsius, the cells stay in a completely inactive state and thus they can be conserved for longer period. Now this cryopreservation has been successfully applied for germplasm conservation of a wide range of plant species like rice, wheat, peanut, cassava, coconut, etc. And several plants can be regenerated from cells, meristem, embryos, etc., stored in cryopreservation. So that's all about conservation method, germplasm conservation techniques. Thank you all.